Our research group looks at neonatal brain injuries, and of one of the streams of brain injuries that we look at is perinatal asphyxia, which is basically defined as a hypoxic-ischaemic event around the time of birth. It's the leading cause of morbidity and mortality in term newborns, and is associated with neurodevelopmental disabilities such as motor and cognitive disabilities and cerebral palsy and epilepsies. So as I said, perinatal asphyxia is a hypoxic-ischemic event around the time of birth, which results in reduced oxygen and blood flow to the baby, usually in the intrapartum period, so during labour and delivery. And it can be, there are numerous causes such as a placental abruption, or the umbilical cord gets tied around the neck, or there's a condition called meconium aspiration, which is when the baby has a fecal discharge during birth and then sucks that into the lungs, and it can impede respiration then once the baby's born. And so this, when there's a perinatal asphyxic event, if the baby develops a brain injury, this is known as hypoxic-ischaemic encephalopathy. And at the moment, there's limited clinical treatments to resolve this brain injury after HI events, but one of them is that's currently in routinely in clinical use is therapeutic hypothermia, and then the symptomatic treatment of things like neonatal seizures. So our interest, my PhD was looking at the GABAA receptor in the newborn brain, and the reason for that is that in the brain you have various neurotransmitters, and GABA is usually part of the inhibitory neurotransmission system in a mature brain. However, in the developing brain, it actually has some excitatory functions, along with your characteristic NMDA receptor neurotransmission as well. And the reason for this kind of precocious excitatory neurotransmission in the brain is it helps develop the neuronal circuitry and the development of cortical connections. So the GABAA receptor is a ligand-gated chloride ion channel, and it will function through binding of the ligand to the receptor, or a equivalent agonist substrate, will result in a chloride flux according to the electrochemical gradient. And this is established through cation chloride cotransporters. So this on the left is a figure from a review that looked at messenger RNA expression of the various cation chloride cotransporters, and in the mauve line you can see that KCC2 is heavily developmentally regulated, with an increase in mRNA expression through the late prenatal and into the postnatal period. And as I said before, while KCC2 still has a low expression, there ends up being higher intracellular chloride than extracellular, such that when GABA binds to a GABAA receptor, the flux of chloride is at an efflux out of the neuron, and that results in depolarization or further excitation. So we've had a brief look at KCC2 expression, the potassium chloride cotransporter 2 expression in the pig brain across development in our in a couple of brain regions, and we've also observed in protein expression this increase in that perinatal period of KCC2 expression. And this coincides with the switch in GABAA receptor expression, function, where it has some excitatory roles into its more mature inhibitory roles, with the increase of the potassium chloride cotransporter that extrudes chloride ions out of the neuron, creating a low intracellular chloride environment. So, as I said before, hypoxic-ischaemic encephalopathy occurs after neonatal hypoxia, and it's the leading cause of neonatal seizures. And as I said, the neonatal brain is vulnerable to seizures due to its hyperexcitable state, with the GABA receptors, some of the GABAA receptors still having excitatory actions, rather than the standard presumed inhibitory neurotransmission. And the concern here is that in neonates, they treat seizures with the same GABA agonists that they use in the pediatric and adult population, and these GABA agonists are targeting the inhibitory action of the GABAA receptor. However, if a HI event or seizures are evoking alterations to the GABAergic system, both through GABAA receptors or these cation cotransporters, then the efficacy of these treatments will be impaired. So some rodent work in juvenile rats by Naylor and colleagues looked at the expression of NMDA receptors and GABAA receptors after seizures, so not after hypoxia. And they've observed that there is a increased membrane expression of the NMDA receptor subunit NR1, and it gets transported out to the membrane, so increase in excitation after seizures. And that in this juvenile rat, when GABA is acting as its inhibitory action, after seizures you get an internalization of the GABAA receptor. So again, that's predisposing the brain to an excitatory state after seizures. After neonatal hypoxia, there's a cascade of injury events, and you can see that seizures occur in the period following a latent post-initial insult phase, and they can occur in the days following, as can other injury processes such as inflammation and excitotoxicity. And so subsequently, therapeutic hypothermia is initiated usually clinically within 6 hours after the HI event has been recognised postpartum, and they usually will treat a neonate for up to 72 hours and then rewarm the baby for 10 to 12 hours back to a normal body temperature. And that's really targeting that therapeutic window of the secondary injury phase post-hypoxia. So what we have is a newborn piglet model of perinatal asphyxia, where we get the piglets at less than a day of age, and we induce hypoxia-ischemia by reducing their inspired oxygen percentage. And then at the end of insult, we either instigate normothermic recovery under a mild anaesthesia, where we keep them at normal body temperature, or they have hypothermia for 24 hours with a 9 to 10 hour rewarm. At this stage, it's at about 44 to 46 hours post-insult. We wean them from their anaesthesia and their ventilation, and then they're recovered through to 72 hours post-insult, which is day 4 of life, and they have an MRI, and then we euthanize and collect the brain samples. So with the MRI, we can, using our newborn pig, the size of the animal, it enables us to perform procedures that can be applied clinically to neonates as well. And one of the procedures clinically they'll use to prognosticate or diagnose the severity of the injury is MRI and the spectroscopy, and we can look at the different ratios of the metabolites. So N-acetyl aspartate is a marker of neuronal survival, and lactate is an indicator of the hypoxic event. Choline is a marker of membrane turnover, and this is elevated in pathological conditions. And creatine is found in metabolically active tissues, and it's a marker of intact brain metabolism as it's involved in ATP generation. So we can assess how healthy or damaged the brain is through this non-invasive measurement and use these ratios. So in this figure, the green squares are the sham animals, the red circles are the normothermic HI animals, and the blue diamonds are the hypoxic animals that received hypothermic treatment. And we can see that hypothermia has significantly improved some of the metabolic state of brain activity in these animals when assessed in vivo. We can also see, looking at the behavioural outcomes and then a histopathology on H&E scoring of the brain regions, that therapeutic hypothermia provides moderate improvement in the behavioural scoring at day 4 of life, and there is some neuroprotection when assessed through H&E scoring on histopathology. So as I said, we were interested in what the protein expression of the potassium-chloride cotransporter 2 was. So we've done that initially through Western blots. We assessed 5 different brain regions, but this is just an example of one of my blots of the parietal cortex. And you can see there are, on this gel, there were 4 sham animals, 3 hypothermic and 3 normothermic. And the commercial antibody is designed to recognise the full-length KCC2 at 140 kilodaltons. And we can see that 2 of the therapeutic hypothermic animals, while having reduced expression, still have a band at 140, and 1 appears not to have the band present in the parietal cortex. And then the 3 normothermic animals appear to have a cleavage band more around the 85-90 kilodalton, a kind of doublet band there. When I looked at quantification of the full-length KCC2 band, the sham animals in the open bars, you can see that there's significantly higher expression in all of the brain regions, and there was a non-significant increase in KCC2 expression in the hypothermic treated animals compared with the normothermics, but across all brain regions this was non-significant. We can then look at the association of KCC2 expression with some of our in vivo and ex vivo outcome measures. And so we can look at the MRS ratios, which we saw significant differences in the group. However, some of them show an association with KCC2 expression, whereas, for example, the lactate N-acetyl aspartate ratio does not. Histopathology appears to have a moderate correlation with KCC2, but when we take out the sham animals, which all have no overt histopathology, we lose the association altogether. That's just a line of best fit, but you can see that there's no relationship between severity of injury and your average KCC2 expression across your 5 brain regions. So I've started looking at the cellular localization of KCC2 in our cohort. And when we look at the expression of KCC2 in the parietal cortex layers, in the sham animal you can see distinct neuropil labeling, particularly through cortical layers 2 through to 4, which includes layer 3 is the external pyramidal cell layer, and then the external and internal granule cell layers of the cortex. And when you then look on the other left-hand box, the moderately hypoxic animal that received hypothermia, It appears to have a similar pattern of KCC2 expression at the cellular level. However, when you look at the right-hand two panels, they are the moderate to severe HI animals. The top one's normothermic and the bottom one received therapeutic hypothermia, and they both have significantly altered KCC2 expression and pattern of expression in the cortical regions. I have co-labeled these with NUN but haven't imaged it, so yes, early days of imaging, but we'll have a further interrogation of that to come. And also looking at the cerebellum, in the sham animal, you can see lovely discrete membrane localization of KCC2 on the Purkinje cells, and there's evident labeling on the cell soma and processes in the molecular layer. Whereas after hypoxia, you can see, particularly in that molecular layer, a disruption to the expression pattern of KCC2. And then in the normothermic animal in the middle panel where the arrowhead is, there's particular, it seems to be internalization of KCC2. So I'll have to look at that at a bit higher magnification yet, and also potentially co-label like Naylor did with synaptophysin, which is a synaptic membrane marker. So our next steps, KCC2's chloride extrusion kind of actions is facilitated by phosphorylation at a serine 940 site on the transporter. And so phosphorylation at this site under normal conditions is determined through protein kinase action, and then it is dephosphorylated at that site through the protein phosphatase 1 actions. And so under normal conditions, there's regular membrane recycling of KCC2 that's endocytosed by the adaptoprotein 2 molecule. However, under pathological circumstances such as seizures and the prolonged exposure to neurotransmitter substances, you end up getting a triggering of higher intracellular chloride through entry by the NMDA receptor, and this triggers or this favours the protein phosphatase 1 action, so dephosphorylates at serine 940, and also there appears to be neurotransmitter receptor dependent changes to KCC2 phosphorylation that are activity dependent through cleavage by calpain, which is a calcium dependent protease involved in the degradation of KCC2. So you end up having extra degradation of KCC2 under pathological conditions on top of the endocytotic recycling of KCC2 from the membrane. So we'd like to look at investigating the presence of the amount of phosphorylated KCC2 using a commercial antibody that targets the serine 940 phosphorylation site, and also looking at calpain expression in this cohort of animals. And just briefly, rodent models have shown that therapeutic targeting of inhibiting calpain's actions do result in stabilizing KCC2 expression on the membrane. So you can see that in the bottom panel from Wan et al, that when they induced seizures in a juvenile rodent and then post-treated with a calpain activity inhibitor, they were able to stabilize KCC2 expression on those pyramidal neurons in the hippocampus. So there's lots of work still to be done. It's half a story at the moment, but I'd just like to particularly thank the Bjorkman group for their involvement with all the lab work and the animal work. And that's it.